just a toxic girl slipping through your fingers. She's gonna ruin your world. Little toxic girl. She's just a toxic girl slipping through your fingers. She's gonna ruin your world. Little toxic girl. You know this girl is deadly. She's poisonous. A single kiss won't kill you, but you'll never be the same. She'll steal your heart from you. She'll beat it black and blue, and you can stop it. She's addictive, toxic. Her venom flows in you. She'll make you sick like glue, and you can stop it. You're addicted. She's just a toxic girl slipping through your fingers. She's gonna ruin your world. Little toxic girl. She's just a toxic girl slipping through your fingers. She's gonna ruin your world. Little toxic girl. She's just a toxic girl. She's gonna ruin your world. She's just a toxic girl slipping through your fingers. She's gonna ruin your world. Little toxic girl. She's just a toxic girl slipping through your fingers. She's gonna ruin your world. Little toxic girl. Slipping through your fingers.
All right, good morning, everybody. Today is the 15th of April, and we're going to be continuing our lesson on measuring evolution. So, remember one of the things that I said before was that we had to, this evolution is a part of science, and in science we have to be able to measure. So, pull up my window. So if you look at this, again, here is kind of my description of natural selection. So you have this landscape where there are mice as the prey, and then you have a bird as a predator. And in this landscape, there are light-colored brown mice and darker-colored gray mice. As the bird flies overhead, which ones is it going to spot? And you notice the proportions in the population, how you have 3 darker colored mice, and then you have 6 lighter colored mice. But then as time goes on, it's going to cause a shift. So now you have those 3 original that are left, but only 2 tan mice make it to or make it past predation to the point where they can mate, because that's the driving force in evolution. Is you have to be able to pass on the traits that allow you to survive better in your environment, because survival of the fittest or fitness does not mean the strongest or the like how we would think about fitness as physical fitness. It's fitness as in your ability to survive in your environment to make it to reproduction and producing viable offspring. So in this example, these 2 tan mice are left over from the original population because they don't blend in with the background. These 3 black mice are able to reproduce, and notice how you have a higher proportion. Excuse me. If this goes on long enough, eventually either the tan mice, that allele, or those traits that allow it to be tan will become extinct, or they will need to develop some sort of behavior that allows them to survive predation. And then as a kind of a feedback loop, this bird will need to find a way to hunt the darker colored mice. It'll either have to look for movement or something, right? There will have to be some sort of feedback that happens. Thinking back to the lion and the gazelle, the reason there are no slow lion is because all the gazelles are fast, and the reason that there are no slow gazelles is because all the lions are fast. So this bird will need to find a way in itself to evolve and overcome adaptations. So today we're going to be, let me pull up my lesson plan for today. So today we're going to kind of continue on part 2. Remember, originally we had talked about how there are, we do comparative anatomy. So in comparative anatomy, we're literally looking at, or with comparative anatomy, we're looking at how do these animals look alike. So looking at the hands or feet of humans, whales, dogs, and I have the notes right here. Dogs and birds, about how they still have the same bones in the same order. It's just their shaping is a little bit different. So whales, underneath their flippers, do have fingers the same as humans do. It's just they're not able to control them in a similar fashion to us. And then also seeing vestigial structures. So the example was tailbones in humans, hind leg bones that are in whales, snakes that have undeveloped legs. So those are all vestigial structures, just leftover structures from our evolutionary past that serves no function. And then we talked about analogous features, which was 2 things that evolve separately in animals. So for example, the example I used was the white fox and the bird. Both of them have white coloring that darkens during the summer and lightens up into white in the wintertime, but they serve 2 different purposes on each of those animals. Then we talked about molecular ecology, using genetics in order to find out different things about organisms. So today what we're going to start in on, it didn't change my color again. I don't know what's going on with this. We're going to talk about biogeography. This is saying how you have multiple things found in different places. So a primary example is are ferns in Greenland. All right. So there are fern fossils in Greenland, even though there are no ferns themselves growing in Greenland. So how did they end up there? Well, I mean, you could propose that, excuse me, they may have been taken over by human settlers or colonization or something like that, left behind, and then ended up in the fossil record. But looking at how the fossil record functions and how old it is, that's not the case. But if you look at Pangea and you have Greenland, Greenland fits nice into another section where there are ferns that are present, and now there are fern fossils over here. All right. And it's thought because these 2 would have been joined together through Pangea that there would have been ferns there, and then as the continent started to drift apart over the years, then they would have become separated, and now you have these 2 areas, once sharing the same biome, now separated by climate, precipitation, all sorts of things, and their biomes are separate, but you see that they share a similarity in species. So a really common one that's always brought up are the amount of marsupials. So here are some examples of marsupials. So marsupials are any animals that they carry their offspring in a pouch rather than through how typical mammals have placenta. So these are non-placental animals. And so then you have to kind of look like, why is there such a high amount of species diversities of marsupials in a place like Australia? Because these animals right here, you look at these and you know koala and kangaroo, the two places that you're going to instantly assume is you're going to go right back to Australia. All right, so Australia's been separated from other places by billions of years, and there's a massive amount of water in between them. And I haven't seen them in a while, but I'm pretty sure kangaroos just don't hop across water to get to where they're going. And so you could think about it similar to Darwin's finches in Galapagos. Some of these marsupials are present in other places other than Hawaii, or other than Australia. Why would they be present there? Well, there was some sort of land connection that allowed these animals to move across, and then instantly, once these barriers started to open up, because remember, it would have been a small stream at first, and then when it would have been a river, and then it would have split apart into an ocean. And it gets to a point, excuse me, it gets to a point where these animals can no longer intermingle, and they now have this geographical separation in between them. And I know that there are other examples besides this, but marsupials are kind of the go-to with it. And they would have been able to evolve, and then they didn't have competition as those areas started to split apart. Because remember, as those areas start to change, their climate's changing. Is it changing rapidly overnight? No. But over the course of a couple million years, you are going to end up with this change in climate, and that's why you tend to have a high amount of marsupials in Australia versus mammals, because there's this biogeographical shift. And a lot of this does have to do with Pangea. You know, having an original supercontinent where everything was joined together, and now it's completely split apart. So pulling up a map, I think we've all kind of seen this before, but pulling up a map, you notice that we always kind of brought up, like, have you ever noticed how South America kind of fits in really nice in here onto the west coast of Africa, and how Madagascar looks like it should kind of slot into this area right here? And same thing, Australia would have been further up here. This entire bit of landmass right here kind of fitting in through this, and then of course the ferns in Greenland kind of slotting into around this area. And then, of course, like, it looks like it lines up on this side. And so that is the idea of this, of Pangea, or a supercontinent, is that all of these kind of fit together like a puzzle piece. Yes, I understand that they don't line up perfect, that there's no one-to-one match, that this shape isn't 100% identical to that shape. But you have to think that over time, with geographical features, and as they're starting to split apart, you know, it'd be like if you were to pull puzzle pieces apart rather than split them like they're supposed to. So rather than moving them vertically, you kind of pull them apart horizontally. There's going to be some damage, and they're not going to fit together 100% perfect. So that is where this whole thing of biogeography kind of comes into play with evolution. Because if there was a species that was here previously, it was probably also here when these two shared a landmass, and now they have an entire ocean between them. Same thing, ferns in Greenland, and all the marsupials in Australia. All right. It tends to be a pretty easy concept to kind of grasp. I think most people have kind of heard of Pangea before, and they've kind of heard of this, a lot of these concepts. So then we're going to get into something that's a little bit more I guess it's not as it's in science we look at this and we understand that it's pretty good and it fulfills its purpose, but it is very controversial outside of science. And that's why General Colin Powell always said, like, don't take stock in your naysayers. So it's one of those things that in science we pay attention to science. And so for the next one is the fossil record. So the fossil record, and I have a picture for this. You can see the different layers in this rock formation. I'm not sure where this rock formation is, but you could see the stratification in it. And each of these layers are called strata. And we talked about stratification when we talked about the tree, about how there are different segments in I want paint. When we talked about in a tree, you have these different layers within the tree, and within these different layers and because I suck at drawing, I'm just gonna so within the tree you have these birds that they feed on different zones within the tree. And is there overlap? Well, yeah, you could see a little bit of overlap right here with I guess I should make it a different color. With these species right here, there is a little overlap. But for the most part, each of them kind of has their own section that they feed in and that they live in, and there's very little competition between them. So we had talked about stratification with this example. So back to looking at the rock. So you have this stratification, and each of these layers is formed over time. So as soil develops, all of this material that's on the top will eventually start to die off as larger plants come in, and that will make up the soil. As those organic compounds are broken down, it will start to move deeper and deeper as more stuff is piled on top of it, and eventually it will calcify and form rock, or whatever other materials are found in that specific type of rock. And so how do we use this to our advantage in evolution? Well, we know that fossils are a thing that exists, that as an animal dies, a small, tiny portion of those animals will end up embedded within this fossil layer. And so let's say you have an animal that dies, and it's in, like, let's say it's in this layer right here. Let me copy this and put it into paint. All right, so let's say, wow, that's very pixelated. Let me adjust it. So I don't need the bird example anymore, so we'll cover that up. So let's say you have, an animal that dies in this layer right here, right? What you could do is, you know that this is the youngest layer, and this is the oldest layer at the bottom. So what you can do is you can actually date this through carbon dating. And I know that a lot of people are like, well, carbon dating isn't that accurate. It is a lot more accurate than what people give it credit for. It's not going to say like, oh, you found a fossil in here, it died, June 8th, and it was exactly, 1,256,922 years ago. It's basically like it died around this time frame. And using that, we could kind of get a rough age of when this lived. And when you're talking about animals that lived 90 to 95 million years ago, is it really all that big of a deal that you narrow it down to the exact date? Because what you're trying to do is you're trying to get a good picture of when they existed. Look at humanity right now. does it really matter that humans existed now? And does it matter that humans existed 5,000 years ago, or they'll exist 5,000 years into the future? So that's kind of what we're looking at, is what was going on during this period of time. And even then, that's way too big of a thing. We're more concerned with like, what does this look like? So then, in order to increase the amount of accuracy within here, yes, we have carbon dating. We have carbon dating, but we also can use xenon and a lot of other elements in order to get a really good idea of how long ago this was. Again, remember yesterday when we talked about how, or I'm sorry, not yesterday, but on Monday when we talked about how there is this community and this collaboration within biology, that my research contributes to somebody else's research, which will then contribute to somebody else. Like my research is I did not come up with all these thoughts and ideas and things like that alone. A lot of what I have is based on things that happened in the past, and I found scientists that did this research and they had openings in their research, and I'm like, oh, okay, here's a question they didn't answer. That's a question that I can answer. And so then I take their work and kind of take it to the next step. Same thing with this. We just don't use carbon dating. There's argon and all sorts of other things that we can use in order to date these different things. And all of those kind of collaborate together to come up with a time frame. And all they do is kind of increase that accuracy, because remember, you're never going to hit the center of the bullseye, but you can get infinitely close to it. And that's what we're trying to do when we do a lot of stuff as far as looking at the fossil record. So fossils are great for looking at creatures that no longer exist. All right, this is looking at things in the past that their entire lineage has been completely wiped out. Yes, we can say, oh, modern day birds are related to dinosaurs, but we really don't have a modern day dinosaur that we can go and observe. There's not like a descendant of a T-Rex roaming around in, Foster Park or wherever. So we use the fossil record in order to see like, okay, what was life like for this animal? And a really, really common one that is used quite a bit is Sue the dinosaur. So Sue the dinosaur is on display at the Field Museum in Chicago. I understand that if I say go to the Field Museum in Chicago, you just can't be like, all right, I'm on my way there now. But this is the largest, most complete skeleton, and it's set up in the lobby like this in the Field Museum. Now, this is not the actual skull. The actual skull is up here in this display case. And just because the sheer mass of this thing is so big that it would be difficult to suspend it. Now, this is the most complete skeleton, but there are some fabricated parts that are in here. And because of this, they were able to do all sorts of studies on this in order to look at, okay, what happened during this animal's life? So when you have fossils like that, so going through this, the death occurred in a seasonal stream bed, which washed away some of the small bones. That's why I said not all this 100%. During life, she received several injuries and suffered numerous pathologies. An injury to the right shoulder region resulted in a damaged shoulder blade, a torn tendon in the right arm, most likely due to struggle with prey, and 3 broken ribs. The damage subsequently healed, though one rib healed into 2 pieces, indicating Sue survived the incident. These are things that we can look at through fossil records. The left fibula is twice the diameter of the right one, likely a result in the as a result of infection. So what did it say? The left is twice. So if you look at the left one is definitely a much larger than the right one. Where was I? Okay. And original reports of this broken bone were contradicted by CT scans, which showed no fractures. Multiple holes in the front of the skull were originally thought to either be bacterial infection or bite marks from other T-Rexes. Subsequent studies show these areas of parasitic infection, possibly from infestation of an ancestral form of, I'm not even going to pronounce the, oh, it's a bird, a protozoan parasite that infects birds that ultimately leads to death by starvation due to internal swelling of the neck. Damage to the back of the neck was interpreted. So continue. You can read this and then go to the field museum and see Sue for yourself. But a lot of this tells us what was life actually like for this animal back in the day. And this is just one T-Rex of, millions that would have existed. And we know a little bit more about it, or at least we know a little bit more about one. And that is, and there's no reason to assume like, oh, well, she's, or her life was a little bit different than everybody else's. You have to kind of assume that if the life of a T-Rex falls upon a bell curve, that she's going to be right in the middle with the average, since most are right in the middle with the average. And it's one of those things that we can use the fossil record and the information that we have in order to find out about organisms that didn't exist. We can also use it to construct family trees. And again, this goes back to when we had talked yesterday about comparative anatomy. But with this, we're using comparative anatomy of organisms that don't exist. So this is looking at a horse, right? So this is a modern day horse. You see kind of the shape of its skeleton. This would be it munching on some stuff. Here is a skull for it, so you can see how the teeth are formed, and then you have that front set of creepy teeth that's on it, and then the formation of its feet. Now, there are some things to pay attention to, so you could pay attention to this ankle bone, and then pay attention to this knob that kind of sticks out, and then just this formation about how it's smooth all the way down to where its nail is. Now, going back, looking at the one of its ancestors. So you can see skull shape is pretty similar, and it's mounted on its neck, which we're going to assume can move the same as what this horse does. Its legs are kind of moving in the same direction. Its back is a little bit flatter, but that could just be because the way that this one is prepared, it kind of shows arches in it. And then how its hip structure is formed. Notice in the skull, this the way that its teeth are shaped, it is a lot more kind of angular with less of a flare at the very end of it compared to in a modern day horse. Notice the eyes have moved further up on the skull, and then this little bony protrusion is slightly smaller. This bulb on its leg has increased in size, while this kind of notch for its ankle has decreased quite a bit in size compared to the one before it. Now, going a little bit further into the past, you can see that it's gotten a little bit smaller, but this might just be perspective of the photo. But you can see it's gotten a little bit smaller. Its hip structure has changed quite a bit. This flared part on its nose is pretty rounded. Notice with the front teeth tend to extend a little bit further back compared to modern horses. Its eye is in relatively the same position, but these teeth tend to form more of a sloping angle, similar to with this previous one. This bulb on its the bulb that's on its leg has decreased again in size. And then notice how now you're starting to see multiple toes. And that is a bigger deal, because there is a distinction between animals that have toed feet and animals that have a single hoof, because that can reflect adaptation to its environment. So again, this question is going to seem a little redundant or it's going to seem kind of directed, but normally I ask this in front of the class, but why would having these multiple feet or these multiple toes be at an advantage over having a single hoof? and think about that more as its landscape. So, which one of these would be better prepared for a rocky landscape? Well, having more toes would help out with a rocky landscape because you're able to kind of spread weight out and find multiple places to grip to. What about this hoof? Well, this will probably be a little bit more suited for more solid ground that's flat and even. It's a little bit better for maybe running. So there are kind of these differences that form. And then going back to very early ones, you can see eyes are very, very far forward compared to what they were in previous ones. It's like front lip area is moved further in, making it look like this little protrusion is smaller. This piece, so this part right here is moved in. Its teeth take up most of the length of its jaw. And then looking at it, it is, I don't know, again, I don't know if this is a matter of perspective of the picture, but it does look like this animal is a little bit smaller. And then you can see that there are full-fledged toes on it, and this bulb or bony protrusion that exists on the modern horses is now gone. And then the feet, there are full-fledged toes that are on it. So think about why that would be an advantage, because remember, there was a Russian scientist who said that nothing in biology makes sense without looking at evolution first. So why does the animal have this? And evolution is kind of the answer to that why. Why is this the way that it is? Well, it's because it has some sort of advantage within its environment. Every year, a student always asks a question, is this a form of adaptation for this animal, or did the animal evolve this way? And the answer is always yes, because whatever that trait is was better suited for its environment. Now, if, let's say, horses eventually start to get pushed out of grassland areas and then end up in rockier areas and things like that, we might see a move back toward toes given enough evolutionary pressure. But again, that requires variation within the population. The problem with horses now is that a lot of times their genetic information is extremely controlled because we've domesticated them. There are wild horses that exist. They're native, or they're not native to the US. They were brought over. But for the most part, they have been domesticated. They are domesticated, meaning that we have bred them and selected traits that we want as humans to have them serve a function for us. So look at your dog, whatever your dog is. If you have a husky, huskies were selected for their endurance. German shepherds for their intelligence. I have a dachshund, so wiener dog. They're selected for their hunting ability and their ability to burrow underground with long, low bodies. And then there's other dogs like, Pomeranians and things like that that evolved for to look at or for aesthetic value. We do breed animals and plants for aesthetic value. We will get into artificial selection later on, because artificial selection does influence gene pool over time. Again, if you guys have questions, feel free to post them into the chat. I know I'm moving pretty quick. So then what we're going to do next is we're going to look at us actually observing. So we have, so this is direct observation. I write like a 2-year-old that's like has full grasp on a crayon and is trying to write. I am terrible with writing with a mouse. So looking at direct observation. So how can we actually observe this? Well, we have a lot of the stuff that we talked about before. We have the fossil record, but those aren't direct observations. Those are observations that we're making afterwards. The same thing with molecular ecology. These are observations that we are making afterwards. So what can we see right now? Well, there are examples of like lizards and things like that we've observed their evolution, but the fastest thing that we know of, the fastest growing stuff that we know of, is insects and single-celled organisms. So one example is that I'm going to bring up because it's one that I know the professor that does the research on this is non-pathogenic tuberculosis. Okay, tuberculosis is a bacteria that gets into your lungs, and as it starts to reproduce, causes pneumonias, and you eventually die from it. It is one of the it is a disease that if you look at the number of people that die from tuberculosis in the US, it's very minimal. Around the world, it's extremely high. It is one of the most dangerous diseases that's out there. So how do you make non-pathogenic tuberculosis? Well, I'm going to draw this in red. So you have this tuberculosis. There's this DNA. Then it's got a plasmid. And remember that bacteria do not like to keep on genes that they don't keep genes on board that they can't use. So then you plate it into a growth medium. Same thing. Then you plate it again onto a growth medium. Get the same thing. Then you plate it again onto a growth medium and get the same thing. And then you plate it again, and it instantly kicks out. I'm gonna draw that. Yellow is a terrible color. Then it kicks out some of these pathogenic genes, so it's no longer infectious to humans. All right, because remember, if a bacteria has that on board, it has to maintain it through replication and protein synthesis and all of those other things that we had discussed previously. So now you have non-pathogenic tuberculosis. This could be argued that yes, this is artificial selection, but it's still a form of evolution that we have observed. Then another thing that we've observed is drug-resistant pathogens. And when I say drug-resistant pathogens, I'm mostly referring to bacteria. All right. So what happens with this is that when you have bacteria, here's your bacteria cell, here's your main set of DNA, and here's your plasmid. All right. Your plasmid, that arrow, your plasmid, as we talked about in when we went over cell biology, your plasmid kind of contains accessory DNA. It's not stuff that's required for survival. It's just stuff that kind of helps out. But let's say you have another bacteria that comes along and in its plasmid, it has a section of it that allows it to be resistant to an antibiotic. So I did this when I was taking genetics in college. We did this with ampicillin, where we made ampicillin resistant E. coli. So now you have this bacteria that has the resistance. So this one has it, this one doesn't. And what they do is then they can link their cells, and they can replicate and pass this thing over and insert it into the other cell. So now you have two that have it. So now both of them, I'm going to correct that to have it. And so why is this dangerous? Why does this happen to be so dangerous? And again, I'm going to go back to an example that I know of. When I was in the military, we used to have a problem with, there was a thing that originally started called Marine Corps Martial Arts, and everyone was certified in it. And you can move up in the ranks of Marine Corps Martial Arts by getting new belts and passing certain courses. All right. So you started off, I think it was tan belt was the lowest and black was the highest. And then you got stripes on your black belt. So like black with a red stripe and things like that. And what it did was it taught you these different techniques. And a lot of them were grappling techniques and things like that, because the hand-to-hand combat was one of those things that had kind of grown out of favor. And so this was kind of a return to tradition. And they had the Marine Corps Martial Arts Program, or MCMAP. And then, of course, Marines, they're 18, 19, 20-year-old, individuals, and they are, they get bored, so they wrestle around in the barracks and stuff like that. Well, one of them ended up with a bacteria called MRSA. That's how you spell it. Methicillin resistant. MRSA, that's it. I had to think of the name of it for a second. So it was methicillin resistant Staphylococcus aureus, and it is a staph bacteria that is resistant to methicillin class drugs. So why is this a big deal? Well, because once one guy gets it, so let's say it's a Marine, so I'll draw him in green. So this guy gets it, he's happy. And then he passes it on to another guy, who then passes it on to another guy. Is it that big of a deal? No, not really. But let's say Marine C here was out doing something stupid. All right. And you guys have heard my stories about how Marines would, they do dumb stuff all the time. So this guy's out doing something stupid, and all of a sudden he gets a little boo-boo on his arm. All right. That's blood dripping from his arm. So he comes and sees me like it's the end of the world, because I was a medic with the Marines. And he comes and sees me like it's the end of the world, and I give him a Band-Aid and some Motrin. And then he goes back and does whatever. Well, now, remember, he's been exposed to MRSA from person to person to person. Now he has an open wound, and after time, what was originally just a small issue, especially if he doesn't keep it clean and he's not up on his hygiene, instantly becomes a big deal. And so now he ends up with a MRSA infection. And you can look up online of what MRSA infections look like, but imagine a pimple the size of a quarter, and that's how MRSA infections kind of play out, at least on the extreme ends of it. They can get really bad. But that happened because this drug-resistant bacteria is being passed from person to person. Now, it's really common that you would give out penicillin or most, like, usually the first thing that used to happen was they used to give people penicillin shots in order to kind of treat a lot of diseases that happen. And that led to these antibiotic-resistant bacterias. MRSA being one of the ones that we had to deal with quite a bit when I was in the military. I didn't see it as much because I really emphasized hygiene on on a lot of the Marines I was with. I also worked in the brig or military prison, and it's one of those things that the prison guards had to look perfect compared to the prisoners. Like they could not slip up at all. They called each other by their rank, even if they were the same rank, just because if they didn't, then that would show some sort of vulnerability or openness to the prisoners that they might be able to exploit. And so it was, they were always up to date on their hygiene. Their uniforms were always clean and pressed, so that they looked there. They would portray this look of intimidation off onto the prisoners, and also not show signs of weakness. And so I didn't have as big of an issue as a lot of other commands did, but it was something that we did have a staff-wide meeting to discuss MRSA and what we can do about it. Because of that, there was a shift in the antibiotics that we used to prescribe from penicillin-based medications to Cipro, so ciprofloxacin type of antibiotic. And all of this resulted from a drug-resistant pathogen that existed, being MRSA. So what else do we have that we can observe? Well, mosquitoes. All right. So mosquitoes, basically the worst insect ever made, they are a pest. And in many countries, they are vectors or passages for diseases. For instance, malaria. Malaria, the part of the life cycle of malaria is it has to pass through a mosquito host. So because of that, we can, malaria is not an issue in the US, but we just don't want to get bit by mosquitoes because it sucks. Like you get this welt on your skin and it itches, it doesn't look good, it's uncomfortable, it's inconvenient. So we came up with a way to kind of take care of that. And so we came up with DDT, which is a pesticide that helps, that kills mosquitoes. It does that by inhibiting protein formations. So we spray DDT, and out of a population, some are resistant, some are not. All right, so blue are non-resistant, red are resistant. So what happens if this entire group is afflicted with DDT? Well, now you have a lot of them that were killed off. These two are left, and their offspring, again, the DDT resistant has to be able to be spread to the next generation. They are able to survive and reproduce. And after a while, you end up with an entire population that is now resistant to DDT, meaning that we have this pesticide that doesn't work anymore. And because of that, you're going to see, so for example, if you have a mosquito population, let's say green is resistant, right? What's going to happen to your green over time? Well, it's going to start to increase. And there is a maximum theoretical threshold that the environment can survive. So we called that carrying capacity. And then let's say orange is non-resistant. Well, as the DDT is applied, you're going to see it die off. Now, is it going to collapse all the way to zero? Probably not. There are probably going to be some that survive either through evasion or other sorts of other forms that they use, and just like not being exposed to it at all. All right, so these are forms of evolution that we kind of observe. Now, are they? Can it be argued that these are forms of artificial selection? Yes, it can be argued that these are forms of artificial selection. But for the most part, this is stuff that we observe in lab settings, but we also see it out in nature. So this isn't like it's something that's just isolated strictly to lab environments or anything like that. So we're going to summarize everything that we went through. So we talked about I'm going to use that. That sucks. So we talked about the first thing was comparative anatomy. So comparative anatomy is looking at things that are similar between organisms and just the slight changes between them. Remember when we looked at the fossils of those skulls of horses, how they look very similar, but they did have their differences in them that made them unique and distinct. So comparative anatomy being, I have these two creatures, how are they related? What can I look for that they have? We also looked at molecular ecology, which is how do genes interact with their environment. So ecology being how something interacts with its environment, molecular being genetics. So how do these genes interact with their environment? Again, back to the mosquito example, those alleles or those genes of the resistant mosquitoes are being passed on to the next generation. Looked at biogeography. So breaking the word down is how does geology, the earth, influence the life side of the world. We looked at the fossil record. So fossils, what can we find out from fossils? What can't we find out from fossils? What are limitations on them? And then we looked at kind of some of the modern examples that we use. Many of these are forms of artificial selection, but we do observe kind of the same things out in nature. It is a little bit different with how everything is kind of observed and takes place in a lab. So that is everything that I have for the day. Close all my windows. But this is, I think it's a evolution is a pretty easy topic to kind of wrap your head around once you get past some of the I guess like the myths or the preconceived notions behind what it is and what it isn't, because there's a lot of misinformation about what evolution is. Again, it's not like I'm a horse, I want to reach the leaves up there, so I'm going to make my neck longer over time, and then instantly you end up with a giraffe. What it is that you have giraffes out there with varying lengths of necks, and the ones that make it through some event, the ones with shorter necks are wiped out, and the ones with longer necks make it. So you have to have that level of variation within it. Same thing with like tree sizes and all sorts of other stuff. Remember, it's the mosquito that's resistant to the pesticide that makes it. There are mosquitoes that are resistant. There are mosquitoes that are not resistant, and you're trying to select against one or the other. Now, let's say we develop a new pesticide outside of DDT, then instantly that DDT resistant gene isn't being selected for or against now. And so then variation can start to expand on that, and then it'll select for something else. That's why with pests, they do something called integrated pest management. So if you take, not trying to sell this course because I'm the one that teaches it, but if you take AP Environmental Science, one of the things that we hit on quite a bit is integrated pest management, which is how do we use these different things in order to try to control pests that are out there. But that's all I have for this week. I will be posting new assignments tomorrow. The assignment that I posted last week, you guys should have all the information you need now in order to finish that assignment up. Get it to me either through email or anything like that. If you need any sort of supplemental information, send me an email and let me know. For the most part, a lot of people are doing really well with completing assignments and getting them sent in. I understand that not everybody has access to a computer. I will be posting the assignments as a Word document on PowerSchool. If you don't have Word on your computer, because I don't have Word on my computer either, download it and then upload it to Office 365, and you'll be able to complete it through there. For some reason, the Google integration, I'm struggling with that. So we're just going to download it and then re-upload that file into Office 365, complete it through there, click the share link, send it to me. Remember that you're sharing the document with me, not sharing for me to edit the document. I don't want to edit it. I just want to look at it, see what you've done. I'll leave comments and notes and things like that on it. If I don't respond with comments or notes, then you did fine on it. Make sure you're checking Gradebook. Stay on PowerSchool every couple days. Anyone who's not seeing these lectures, anyone who's not getting onto PowerSchool, anyone who's not checking their email, make sure you let them know to check it, because I'm not the only one. I'm the only one that I think is streaming on YouTube, but I'm definitely not the only one that's still teaching. I'm definitely not the only one that's posting assignments and posting stuff onto PowerSchool. So make sure that you guys are up to date on that. Oh, my recommendation for today. We talked about Sue the dinosaur. There's a documentary called Dinosaur 13, which is behind the process of how they found Sue and this entire process of how they ended up getting arrested and charged with a felony, and it ended up in the Field Museum. It's kind of this whole story behind how that all happened. Really, really interesting. I saw it was on Netflix. I don't think it's on Netflix anymore, but it is out there. I'm sure there's a copy on YouTube that is 100% legit, and I know you guys will get it from a legit source. But it's really good. But yeah, that's pretty much all I have for today. Go to my ending screen. If you guys have any questions, send me an email. But other than that, thank you for watching.